Hello everybody. Over the next few minutes we will talk about the image characterization of untreated bone metastases. We will start off with a recap of the processes involved in metastasis formation and the biological mechanism responsible for osteoblastic and osteolytic metastatic lesions. And then we will spend most of our time talking about the subclassification of metastatic lesions according to their appearances on the MRI scans and how these relate to the underlying biology. In a previous lecture, we explained how tumor cells end up in the marrow space and then hone towards the endosteal surface, eventually displacing the hematopoietic stem cell in the osteoblastic niche where they remain dormant for some time. Once they become activated, they begin the process of colonization, and this process of colonization then leads to this vicious expansion of metastatic disease that is adjacent to trabecular bone. This vicious cycle of malignant bone expansion starts with tumor cells initially signaling osteoclasts via the rank OPG mechanism. The osteolysis releases growth factors and calcium, which causes tumor cells to multiply. Later on, a tumor bed is formed by the signaling of tumor cells via the Wnt pathway of osteoblasts. Osteoblasts themselves lay down woven bone, and it is this woven bone that becomes mineralized with hydroxyapatite crystals, leading to this sclerotic appearances. So to summarize, osteoclastic recruitment and activity causes osteolytic lesions to be formed, growth factors to be released, causing tumor cell proliferation, and this is the vicious cycle. Osteoblastic recruitment and activity causes the laying down of woven bone. The woven bone becomes mineralized due to the deposition of hydroxyapatite crystals. So when you undertake a multiparametric examination of the bone marrow, there are a number of patterns that are visible. Not all patterns are seen at the same time. So in this particular instance, if you look at the left iliac bone, you'll see this has a typical ground glass appearance of woven bone. And then there are metastases that are invisible on the CT scan, but you can see them here on the diffusion sequence and on the fat fraction images. So in this particular instance, we've got two types of bone deposits: invisible deposits and a visible deposit here in the left iliac bone, which has a ground glass appearance. So on CT, we often describe these patterns as being either osteoblastic, osteolytic, or mixed. But now we know that you can have metastatic deposits that occur within normal marrow. So when you look at the dense osteoblastic deposits, we have a range of appearances from the dense osteoblastic lesion to the woven bone type appearance. However, if you start classifying these using MRI, then it's possible to divide these metastatic deposits into other classifications. So you will find that you will have osteoblastic deposits which are of low cellularity, and in fact you will find osteolytic deposits which are also of low cellularity, either containing fat or fibrotic material. And on the other side, you have the more higher cellular metastases, which can be osteolytic, which can be normal, which can be of woven bone type or have a mixed pattern. So it's possible to classify metastatic deposits depending on their MRI appearances into low cellularity and high cellularity metastases. Now let's talk about the highly cellular variants of metastatic disease, starting with the normal cellularity metastasis. Now, all of us have seen metastatic deposits within normal appearing bone marrow. And of course, because there's no woven bone being formed, so the CT scan will be negative. And remember, the woven bone is the target for the bone scan agent, and therefore the MDP bone scan will also be negative. And here is another example. So if you take this patient here with lots of metastatic disease visible on the diffusion image, on the T1 and the T2 sequence, you'll see that the bone texture is completely normal. So in this particular instance, the tumor is proliferating within the bone marrow space, not causing any osteoblastic or any osteoclastic upregulation, so we don't see osteolysis and we don't see osteosclerosis. Another pattern is the pure osteolytic pattern. So here's an example of a pure osteolytic pattern where the bone scan is negative because there is very little in the way of osteoblastic upregulation. Classically, this occurs in multiple myeloma. We've all seen it in renal cancer, but lung cancer and breast cancer, you will also see it. Here's an example in breast cancer. You can see that the bone scan has been read as negative. There is immense infiltration on the T1 and the T2 sequences, which you can also see on the diffusion sequences. And if you look at the CT scans, we can see multiple areas of osteolysis, but very little in the way of an osteosclerotic response, as a result of which the bone scan is also negative. So in this particular instance, you've got tumor within the bone marrow space stimulating osteoclasts, leading to osteolysis, but very little in the way of osteoblastic activity, so you get very little in the way of osteosclerosis. So the right-hand side of this pathway is more active than the left-hand side of this pathway. The classical pathway is where both osteoblastic and osteolytic lesions are active, and this is called the mixed osteolytic and osteosclerotic pattern. And this is the pattern that all of us are used to seeing on a daily basis. So we can see scattered bone marrow metastases on the bone scan. We can see a large amount of infiltration on the MRI scan. And when you do bone radiographs, you can identify areas of osteolysis, and there are areas of osteosclerosis. So in this instance, both the osteoclastic and the osteoblastic activity is elevated, but not necessarily synchronous, as a result of which you get osteosclerosis and osteolysis happening at the same time. The last pattern that I want to discuss is the metastasis with a cellular ring or halo. And here is an example. This patient has metastatic prostate cancer, and what you notice are a number of rings around metastases that you can see on a stir sequence. If you look at the center of a particular metastasis, you'll see that the lesion is in fact mineralized. If you look at this metastasis, you see another halo, and that too is associated with mineralization. Again, you see the same thing here, a halo that is mineralized. So let's take a closer look at this metastasis. If you go axially through this vertebral body, you will see that there are in fact 3 metastatic deposits. There's a metastasis that's anterior and one that's posterior. And you'll notice that the posterior one only has a small amount of mineralization within the center, whereas the more anterior deposit has a greater amount of mineralization. And when we look at the bone window, we can see this smaller amount of mineralization at the back and more mineralization at the front. But you'll also notice that there is a halo around this metastatic deposit. And that is high on the high p-value image and low on the ADC map. So that is a ring of cellularity around a nidus of calcification. Similarly, for the more anterior lesion, you can see it's not as bright. It's not as bright because there is more mineral in this metastatic deposit, and the halo is in fact visible but thinner. So in this particular instance, both sides of the metastatic cascade are going on. Within the center of the lesion, here and here, you've got woven bone being formed because you've got osteoblastic recruitment and of mineralization within woven bone, whereas in the margins you've got delamination of the marrow with expansion of the tumor cells into the normal surrounding bone. And you can show this experimentally. So here's a rat skull on which there is a tumor growing. You can see on the inside there is woven bone and osteoblasts within the woven bone. Here is the cellular ring, and the osteoclasts are seen to be eating away the cortical bone. And it's between the cortical bone and the woven bone that you have the growth of the cancer cells, which on the diffusion imaging you can pick up. And you know that this is an active growing area of disease because you can see these rings expanding. So for example, if you look at this metastatic deposit here, you can see how it grows over time. You'll see another ring appearing here in this metastatic deposit. This ring appears here and then grows. When we look at the diffusion sequences, again, you can see the development of a focal area of hypercellularity that then grows, and look, it's dark in the middle. It's dark in the middle because there is central mineralization of the osteoid within the center of the metastatic deposit. The low cellularity metastases are the osteoblastic lesions, lesions that are lytic but have fat content, and lytic with a fibrous content. So let's take the example of the dense osteoblastic lesion, such as this lesion here on the left-hand side. This lesion is often very radiodense with a sharp margin and is often found to be bone scan negative. And here is an example. So you can see this patient has multiple lesions visible on the T1 sequence, but her bone scan is negative. The whole body diffusion scan is negative both on the coronal as well as on the MIP projection. But you can see that these lesions are visible on the T1 sequence, and when you do the reconstructions, you can see this densely sclerotic lesions with sharp margins. So in this particular instance, we're talking about the excessive activation of the osteoblasts, and that leads to these osteosclerotic lesions, but there's no detectable osteoclastic action visible. Next, let's look at the osteolytic lesion that is fat-filled. And here's an example. This is a patient with multiple myeloma. If you look at the CT scans, what you notice is there are multiple lytic lesions in the lumbar spine. But when you look at the T1 and the T2 sequences, you'll notice that there is no infiltration visible. And on the diffusion sequence, there's no hyperintensity. And this is a very typical pattern of the osteoporotic variant of multiple myeloma, with the space between the myeloma cells being filled mostly with fat. So why do you get fat in osteoporotic multiple myeloma? Here we need to go back to the knowledge of the cellular interactions that we talked about before. The key cell here is the mesenchymal stem cell. Mesenchymal stem cells can become adipocytes, chondrocytes, fibroblasts, osteoblasts, lining cells, endothelial cells. It is the common progenitor cell that can either divide into adipocytes and osteoblasts that is important for this particular explanation. Multiple myeloma produces a protein called DKK1, and DKK1 blocks the Wnt pathway. So you'll see the Wnt pathway is a blocker of osteoclastic activity, and if you block that, then you have free osteoclastic osteolysis. You'll also notice that the Wnt pathway is involved in the maturation of osteoblasts, and that too is blocked. But you'll also notice that the Wnt pathway normally stops the maturation of mesenchymal stem cells towards adipocytes. Now, if you block this pathway and you take away the brake, then the mesenchymal stem cell becomes adipocytes. And that is what explains the increased adipocytes together with osteolysis in multiple myeloma without any remodeling from osteoblasts. Next, let's pick up the pattern of osteolysis with fibrosis, which is this third pattern here. In this case, there is a diversion of mesenchymal stem cells towards fibroblasts and an increase in osteoclastic activation, resulting in osteolytic lesions. So here's a classical example. This patient has breast cancer. You can see that there are areas of metastatic disease that are full of bone, but here you'll notice an osteolytic lesion. Here is another lesion with an area of sequestrum, bone sequestrum in the middle. And here too you'll see that there is some bone sequestrum around this metastatic deposit. Now we would expect the entire lesion to be hypercellular, but that's not what we see. We see that the lesion is cellular just on the margins, so along the margins here, but not throughout the lesion, and that's because the center of this lesion is full of mesenchymally derived fibroblasts. And here's another example of a patient with breast cancer area. You'll see major areas of osteolysis in both the sacrum and in the posterior part of the iliac bones. In this osteolytic lesion here, we've got a little bit of bone, which we see over here. So this is highly cellular, associated with osteolysis. Whereas if you look over here at this metastatic deposit, you'll notice that there are two areas here which are hypercellular, but the rest of this metastasis is not hypercellular. So at the back here, you've got high cellularity. Elsewhere, you've got low cellularity. And in fact, you find the same thing happens over here. So look at this metastasis. You'll notice that it's highly cellular along the anterior margin at the back here, but there is less cellularity associated with this metastasis. And similarly, this metastasis here, which is osteolytic, has low cellularity. And this is because these areas of low cellularity are filled with fibroblasts. And fibroblasts, as we've already said, are derived from mesenchymal stem cells. Remember, mesenchymal stem cells can give rise to osteoblasts as well as adipocytes. And fibroblasts form major components of the extracellular matrix of many cancer types. Okay, let's conclude there with three major learning points. Firstly, there are complex and balanced signaling mechanisms between cells derived from mesenchymal stem cells and hematopoietic stem cells within the normal marrow. Activated tumor cells within the bone marrow co-opt these cellular mechanisms to create microenvironments for themselves, enabling them to proliferate. The use of CT scans and MRI scans allows a new way of classifying bone metastases according to the underlying cellularity, and by looking at the observations, it is possible to find out what are the major signaling mechanisms that are active in any particular metastasis. I hope you found this lecture useful and insightful. Leave me some comments. Thank you very much.